let's move on to the next part in this lecture. We have to know why statistical analysis is important in stability study. Okay, now I have explained to you in brief about what is a formulation, why stability study is important, and now we have to understand why we have to conduct statistical analysis in a stability study. Okay, now when you conduct a stability study, you will have a result or a physical property of the product before and after the study. Okay, so you want to know if these changes or any changes that happens to the product are significant or not. If the changes seems to be insignificant, but after the analysis is shown to be significant, so it is an important thing that we have to pay attention to. So through this kind of statistical analysis, we will be able to determine the suitability of excipients and their effect on product stability. So as an example, product A has excipient 1 and product B has excipient number 2. So you formulate the product and you put them in the stability chamber. And after certain storage time, you take out the sample of the product and you test the product. The result that you obtain can be analyze and compare if the difference between the two or the difference in the physical property of the two products are significantly different or they are same. Okay, so in this way you will know if one excipient is better than the other or there are no difference. Number 2, we can determine if the product characters change significantly over time during storage. Okay, as an example, the chemical ingredient or the API. Let's take an example, the paracetamol suspension. So you formulate a paracetamol suspension that contain 100 milligram of paracetamol in each 5 ml. And then you place the product in the stability chamber at 40 degrees Celsius for 75% humidity. After 6 months, you take out the product and you analyze the content. If the content remain the same, which is 100%, which is 100 milligram in 5 ml, or changes 5% changes plus minus, this is considered as acceptable. But if the content of the API reduced significantly after storage, you would want to know if the changes is significant or not. If they are not significant, so the product can be considered as stable. And the third reason, we want to prove the stability of the product for commercialization purposes. As I mentioned earlier on, when you want to register a product, you would have to demonstrate that the product is stable. So to prove that the product is stable, so that's why we have to have the numbers and the data that say that the product is stable and the changes in the product during storage are insignificant. And another application of statistical analysis is to determine the expiry date of product under development. So through this statistical analysis, we can actually determine the expiry date of product. So I will give you some example of stability study. Now you are developing a nanoparticle formulation containing rifampicin. So what is rifampicin? Rifampicin is an anti-tuberculosis drug. It is highly unstable. It is sensitive to light. Okay, it is sensitive sometimes to moisture. Okay, and it is red in color. So since it is sensitive to light, the container and the closure has to be dark color, or we call it amber color. If the container is clear, okay, clear container, clear plastic container, the stability of the product or of this rifampicin drug is will be greatly compromised. Okay, so there are several factors that need to be evaluated to ensure the stability of the formulation. So the physical characteristics, we have to make sure that the nanoparticle size, the physical appearance, the pH of nanoparticles after reconstitution, and the order has to remain the same during the stability. So this is the physical. Physically, it has to remain the same. Chemically, the API content has to be maintained. The crystallinity means that the drug, if it's in the crystalline form or is in the amorphous form, it has to maintain as it is. Okay, there shouldn't be much changes. And the chemical interaction of rifampicin with the excipients in the formulation has also to maintain. So these are the chemical parts. The microbiological part, we have to make sure that the microbial growth are minimal or absent. Okay, we wouldn't want any microorganism to grow during storage. So what statistical analysis that we can do? So pre and post storage. Okay, before storage and after storage, we can characterize the nanoparticle size, whether they change significantly, the pH of the product, and also the API content. So how do we test this? If we have two characterization, what I mean is that the nanoparticle size before storage is, let's say, 200 nanometer, and after storage it become maybe 220 nanometer. So how do we know if the changes is significant? This can be conducted through statistical analysis. And what type of analysis? Example is PET t-test. Okay, PET t-test is example of test that we can conduct to know if the changes pre and post are significant.